Welcome back to Unit 3, Lesson 6. Today we're going to look at DNA technologies. And as the title says, a little knowledge can go a long way. Perhaps in grade 11 or even this year when you were learning about the structure of DNA, you might have been thinking, why do we need to know it at this level of detail? Well, what we're going to look at today is how that knowledge has been applied to do some cool things. And our goals are to be able to describe the various DNA technologies and their purposes, including looking at PCR, site-directed mutagenesis, recombinant DNA, and gene therapies. And what I'm going to introduce you to here are just a few of the technologies that are available with DNA. So the first one we're going to look at is, what is PCR? PCR stands for polymerase chain reaction. And this is a technique that is used to make copies of DNA in a lab. And you might wonder, well, what do we need to make copies of DNA for? Well, we use them to study DNA samples. So if you have a very tiny sample of DNA, you're not going to be able to do many investigations on it before you destroy it or use it all up. So instead, the first step anytime you get a small sample of DNA is to do a polymerase chain reaction to make a whole lot more of it. So we might be studying DNA samples from crime scenes, from things like amber, think Jurassic Park, fossil samples. We might even be looking for paternity or ancestry. Okay, now it's called a chain reaction because it's like dominoes. Once one falls, they all fall. But really, it's exponential. So what happens is you start with your sample DNA. And you put it in a machine called a thermocycler. So you place it in a test tube, which is going to have some components in it that we'll talk about. And then in a thermocycler. Thermo for temperature, cycle, well, it's able to switch between various temperatures. And in step 1, you denature sample DNA. And the way you denature it, you break it apart into its two separate strands, is you heat to approximately 90 degrees, somewhere between 90 and 95 degrees, for approximately 1 minute. Just long enough and hot enough to break apart the hydrogen bonds between the two strands without actually breaking apart the bonds that hold the nucleotides together. Once you've done this, you go to step 2, which is called annealing. Annealing is a fancy word for sticking together. And what you do is you add flanking primers. To the area you wish to copy. what a primer is. Now they've shown it simplified here, but the red represents the primer. And it is about 20 nucleotides to 30 nucleotides long that are complementary to your template DNA. So if you're actually going to do PCR on some DNA, you do need to have some idea of what the sequence is. But we can do this in humans because for human DNA, we all have some common sequences that we can target. And that primer is necessary for the polymerase to actually bind and work. So that's step 3. It's called extension. Oh, I forgot to tell you the temperature over here. Annealing, for those little primers to bind, you have to cool down to about 50 degrees Celsius for 45 seconds, approximately. It's kind of like making cookies. Every recipe has a slight difference to it. So if you cool down to 50 degrees Celsius, it's enough for the short little primers to bind in, but not cool enough for the entire DNA double strand to come back together. Then for extension to occur, you have to add nucleotides. So you can base pair, and something called Taq polymerase. Okay. And then you have to warm it back up to approximately 72 degrees Celsius for 2 minutes. A little longer depending on how long a sequence you're copying. And what happens is you get your new strand copied, complementary new strands. Which you can then repeat the cycle. They now become templates, samples, and you get exponential growth of your DNA. Increase in DNA sample. Now, you always see them talking on crime scenes about preserving the DNA evidence, not contaminating it with their own DNA. And the reason that's so important is if you have a little bit of your own DNA in here, it's also going to be expanded. Okay. And the idea here with the polymerase chain reaction is approximately within 2 hours you can go from one DNA strand, or very few DNA strands, to 68 billion copies. And then you can take a bit of your sample and hold it aside so that if you ever run out, you can just PCR it again and take all the rest and use it for whatever other experiments you want to do. Now, there's a really cool story here about the guy who discovered this or helped this occur. His name's Kary Mullis. And he won the Nobel Prize for this process in 1993. And really, what he's famous for is he discovered Taq polymerase. Normally, most enzymes, if we added it up to heated it up to 72 degrees, they would die. And in the past, they were using enzymes from humans or mice and using a polymerase that as soon as they heated it up to denature the DNA to 95 degrees, they also denatured the Taq polymerase protein. So it meant every 5 minutes or 10 minutes you'd have to come back and add more polymerase before you could do the next cycle. You can imagine this took a lot of time out of scientists' lives. But Kary Mullis, he was an outdoors guy, and he was out hiking in Yellowstone National Park where there's lots of thermal vents and heated pools, and he was looking at the pools one day and noticed that there was all this sort of sludge growth around it. And he realized that that was bacteria that was able to live in those hot pools, and he thought, oh, they must be able to replicate their DNA, yet they're able to do it at a very high temperature. And he summarized that, hey, their enzymes must be able to withstand really high temperatures. So he gathered a sample of the bacteria, took it back to his lab, isolated out the enzyme Taq polymerase, and then started using it in this process. And it meant now he could just dump in the Taq polymerase and heat it up to 90 degrees and it wouldn't be destroyed, and he could walk away. He liked hiking. Well, now he could set this up and leave it for hours at a time, and it would just run on its own, and he could go off hiking or do other science stuff. So he became the hero of the science world because he freed up many scientists' time. And it's called Taq polymerase because where he found it, it sounds like a bad Roadrunner coyote name, but because he found it in hot water, he called the bacteria Thermus aquaticus for hot water bacteria. And then its polymerase got abbreviated as Taq polymerase. Okay, I'm just going to stop the video here and we'll pick up the other technologies in part 2 of the video.